So we usually have this time, we have an invited speaker. And I just think, because our own seminar of the Blumberg Institute, we actually just not open, even to the residence company here. I think, a lot of people that actually talk about, they actually don't know what we are doing. I think, I will gradually arrange our faculty and participate our, seminar series. So these efforts that will be part of our communication with resident company and also outside the world. Okay, and I was really, encouraged that, during the pandemic, because a lot of the international meeting was stopped. So I started Blumberg webinar and continue our seminar and invite the seminar. That's very good in that 2 years. So right now, I just think, I want, because some friends online, we just want to say that, our Blumberg seminar also welcome outside people, communicate with me so we can actually arrange to participate as a platform for the hepatitis B research work. So today, I just started to give a brief overview about highlights about the research program in Blumberg Institute. So basically, we have total about 12 faculty, and our work involves about 3 programs. One is virology and viral pathogenesis, that's led by myself. That our work is really try to understand how HBV replicates itself and how the host immune response controls virus infection. And we also work on some of plasmid RNA virus like flavivirus, coronavirus. In that case, we mainly try to understand viral RNA replication and innate immune control of the virus replication. And we also have a drug discovery and development program co-led by Dr. Jianhong Chang and Dr. Yanming Du. And we work on the 3 areas. One is targets HBV. In that case, so our goal is that we continue to work on capsid assembly modulator. And currently we have very potent capsid assembly modulator that have a very good in vivo efficacy and PK profile. So that's we are trying to continue the preclinical development, and further work is going on try to get the best-in-class capsid modulator. And we also have the efforts try to directly target the cccDNA and also virus particle and some of our particle secretion inhibitors. We also work on the innate immunity modulators. And Dr. Yanming Du have actually designed a compound that potently inhibits hepatitis A virus RNA replication. And he collaborates with Stanley Lehman at UNC, currently develops this drug. It's a very and in vivo anti-efficacy have already been demonstrated. And currently, with future supports that he will continue those preclinical developments. And finally, we have really a first-in-class yellow fever non-structural protein 4 inhibitor. And about his unique mode of action, we just recently published in PNAS. And Dr. Chang and Dr. Du lead the effort to do the preclinical development, and hopefully in next year, we can hit in phase 1 clinical trial. So the final program is the precision medicine and tumor biology. And the 4 faculty work on those, try to develop assays use cell-free DNA or RNA to provide early detection for liver cancer. And Dr. Pastel's group work on the tumor biology and develop several anti-tumor drugs. So today, I just focus on hepatitis B virus cccDNA biology. And here, this is the most updated fact sheets from the WHO. So despite currently we do have very efficient vaccine and still have about 250 million people currently infected by hepatitis B. And that can lead to about 1 million deaths annually. So what's the hepatitis B? So hepatitis B is caused by a very small virus called hepatitis B virus. So this virus have a very small genome, only about 3.2 kb. And this genome, using the overlap open reading frame, can code it totally about 8 viral protein. So the virus is the envelope virus, is a capsid wrapped polymers and genomic DNA inside, and then outside enveloped by lipid bilayer stamped with 3 viral envelope protein. So when the virus infects the cell, so they're deliver their DNA into the nuclear. So this circular DNA and form a mini chromosome. We call the covalent closed circular DNA. This mini chromosome is the transcription template to transcribe all the viral RNA and then translate into viral protein. And one of these viral RNA that will recognized by a viral polymerase bind into the RNA and then recruit capsid protein to wrap it into a capsid and viral RNA. So then in this capsid, reverse transcribed into a single strand, then double strand. So now you can see this is a very unique virus. The DNA genome is replicated like retrovirus through the reverse transcription. And then this capsid can be enveloped and secreted out as a viral particle. And in addition, the thing is pretty interesting that the viral capsid protein, they can self-assemble into empty capsid. And this actually, in the virus infected cells, more than 90% of those capsid is actually empty. And then they wrapped out and secreted out from called incomplete viral particle, or we more convenient, we call the empty viral particle. And in addition, so the viral envelope protein, in addition to wrap the capsids to secrete out as a viral particle or empty viral particle, they can actually assemble into either small sphere or filaments, lipid particle, and secrete out. So in virus infected cells, you can see that in virus the blood, in the virus infected blood, you can see this viral-like particle, either as a complete particle or empty particle. It's actually a small fraction. The most majority is this sphere or filament particle. So why this virus do this, makes this all this waste, right? Because the biological basis is not so clear, but I think that people thinking this outnumber these particles that actually as camouflage to serve as a decay, to cheating the immune system and helps infection to become resistant. So, because we're going to talk about cccDNA, and you can see that the cccDNA is actually made from viral particle or capsid derived this DNA species, right? So then let's look at what happened in the viral particle. That's pgRNA or pregenomic RNA, how that's actually reverse transcribed into a DNA. So, first, this viral polymers binding to the RNA. This RNA in the they have a stem loop structure, and in the 5' after binding, they can actually find monostrand DNA synthesis. After only synthesized by 3 or 4 nucleotides, they jump into the 3' this region and continue the reverse transcription until the end. That this is how the first strand, we call the monostrand, is made. And at this point, so they have 2 options. So because the reverse transcription, when they reach the end, the last 18 nucleotides, that RNA, cannot be degraded by polymers associated with that RNase activity. So this RNA will translocate to this 5' of the monostrand and initiates the second strand synthesis. And then genome, because here and here have redundant sequence, they jump over the gap and then circularize the genome, makes this called a relaxed circular DNA because there is a gap. Okay. So this is a majority of our genome called the rcDNA or relaxed circular DNA. In the rare condition, that's this RNA primer will not jump and in situ initiates a second synthesis. So then you make a double-strand linear. In viral taping cells, you can majority see this is rcDNA, this is a linear, okay? And you see this mirror, that was actually intermediate, okay? So this is a very unique situation. And then we consider about how the cccDNA is made. So then we have 2 kind of precursor. One is this cccDNA, and another is that double-strand linear. So for the cccDNA, over the years, several labs, especially my lab, Hepatitis lab, and Dr. Jianming Hu's lab, have made a lot of efforts and try to identify the synthesis pathway. So the end product, the cccDNA, is closed circle, okay? And we initially in the infected cells, we found this species that's just like they removed this terminal protein or viral polymers. We call the deproteinized rcDNA. Okay, and then Dr. Jianming Hu's lab discovered that there is an intermediate. The monostrand is already sealed, and the second strand still not sealed. So we call the monostrand closed circular DNA, and then finally the cccDNA. So there's another possibility is that many years ago, Dr. Summers and Mason have found in the PHBV infected ducks and the woodchuck infected woodchuck hepatitis virus infected liver. There is a DNA species called have terminal repeating double strand. The most likely this species is actually synthesized by extending this plus strands into the end, and then extend this by polymers, and then you make this double strand linear. So this double strand linear can actually, either circularize, become a defective cccDNA, or can make a perfect cccDNA through the homolog recombination. So how actually, those double-stranded linear DNA or rcDNA convert into cccDNA biochemically? Let's first focus on this double-stranded linear DNA. So people generally believe that this double-stranded linear DNA, they can just self-circularize. However, it's not that simple because in the monostrand 5' there's a polymer still covalently linked there, and in the plus strand 5' there's the RNA primer still there. So you need to have some end trimming and then ligase. So many years ago, when the Dr. Chong Xiaoxu worked in my lab, he established a stable cell line, only replicates this, produce this double-stranded linear without relaxed circular. Use this cell cut-inducible cell system. We can see that after remote test, first viral pgRNA produced, and then capsids inside the capsid, this DNA replicated, and first monostrand, then double strand. We can see we only have double strand linear, don't have some relaxed circle. And then in the cell, we found that there's a protein removed or deproteinized, this double strand linear. So this already removed, and then you start seeing the cccDNA gradually accumulates. And then he continued, did a self-fractionation study, and found that the deproteinase, that rcDNA, first appeared in the cytoplasm, and then both cccDNA and the deproteinase double-strand linear accumulate into the nucleus. And biochemically, how this happens. So we predict that should be a non-hormonal and joining DNA repair pathway with the final ligation. Okay, to confirm this, we have the Q80 deficient cell line, and then we transfect this the wild type virus in. We always can see that cccDNA can be made, right? But in this defect cell, if you transfected the double-strand linear precursor in replicates that you can, in the wild type cell, you make the cccDNA, but not in the deficient cell. And Hepatitis then did the experiments and transfected supplements this Q80 in this cell. Now they can make a cccDNA. So that perfectly demonstrates this really goes through this double-stranded, non-harmful and joining pathway. And then recent years, people have been noticed that HBV DNA can integrate into host chromosome, and that can actually contribute a lot of the HBV surface antigen production and contribute to the HCC oncogenesis, and also become a barrier for the HBV cure because after decades infection, it's more than 80% of hepatocytes more or less have an integrated copy in their chromosome. How this integration occurs, so that is not this double rcDNA. The main precursor is actually the double-stranded linear or this double-stranded linear with long-term repeats. So mainly from the regular double-stranded linear. So when your chromosome have a double-strand break, this linear DNA can integrate, despite in a very low frequency. However, if the chronic infection lasts for several decades, you can really accumulate a lot. Okay. So then we just talk about the double-strand linear, how that makes covalent closed circular DNA. And then we back to the rcDNA, how that works. So over the years, people have been working for almost 20 years. And I think, the first thing that's very impressively, figure out and by the Dr. Lei Wei. And when he worked with Dr. Klaus in Princeton University, and established in vitro biochemical assay system. And he made this artificial relaxed circular precursor, and then use a cell nuclear extracts, and put this precursor in and can convert into cccDNA. Then he reconstitutes this system and found that you only need 5 cellular protein to convert this rcDNA into finally cccDNA. How that happens? So to seal this gap in the monostrands, so you need FIN1 that is flipless that cuts this flip DNA, and then the ligase 1 is sealed. But for plastrams, they actually recruit 3 proteins that actually catalyze the host cellular DNA synthesis, that ligand strand DNA synthesis components. So they call the replication factor C and the pcDNA. This is the CLAP protein. So, and then recruits polymerase delta to fill it up, and then the FIN1 remove that flipped RNA, and then ligase 1 seal. So finally, this strand finished, this strand finished, and form a perfect cccDNA. However, about 20 years work from many labs, including my own lab, and we have identified inside the cell how the rcDNA to make the cccDNA looks like much more complicated. We either use the gene knockouts, or we use the chemical inhibitor validated by gene knockouts and identify all these protein that's somehow involved in cccDNA synthesis. And especially from the rcDNA, how to remove this terminal polymers. That's someone that is tyrosine, is a tyrosine phosphatase that can actually remove this protein. But that was actually debating. And how from this, how the monostrand is sealed. So those protein seems involved. And then how this happens is not clear, but generally, all this protein looks like involved in the, some of the steps. And recently, Dr. Hepatitis lab have discovered that previous people thinking that this flap will be cleavaged by FIN1, and actually, no one actually sure that this flap is 5' flap or 3' flap. And now Hepatitis found that both the the FIN1 for the 5' flap and this another nucleus for the 3' flap is required. Okay, so that's very nice biochemical detail. We can figure out that. However, for other ones, just like this step, the FIN1 now we know may involve this clap, and the ligase 1 or 3 can seal it. But for other steps, so we actually cannot assign this identified host protein, which exact biochemical reaction they actually catalyze. So in addition, we know that the cccDNA can be formed when the virus infection and delivers a virulent particle that's binding to the receptor. Virulent particle is endocytosed, and then somehow the viral protein interaction with the endosomal membrane and fuse, and then release this capsid in the cytoplasm. And then the capsid docking on the nuclear pore complex to release this rcDNA and get into the nuclear and then form CCC. They also have a pathway we call the intracellular application pathway. For already infected cell, when they make this rcDNA inside the cell, of course, mostly they just envelope and secrete out as viral particle. However, some of those intracellular RC-containing capsid can encoding and release this deprotein rcDNA into the nuclear to make cccDNA. So my lab have found that it's very ironically that cellular factor required to make this cccDNA from the de novo infection pathway and intracellular pathway. Something is shared, something is different. For example, it's found that pol kappa is required for de novo infection, and pol delta also required for de novo infection to fill the gap and form the CC. But for the intracellular application pathway, Liu Li here, when he worked as graduate students in my lab, and discovers the pol alpha is required for the intracellular application pathway. Okay, and he also found that a heat shock protein is required for this pathway, but not de novo pathway. And so why is this? Because one possibility is this, for the de novo infection, we know that when the second strand synthesis still not complete, so this particle already enveloped and secreted out. So when this infected and this DNA precursor, that second strand, that gap is much bigger. So then they may need a different repair path. However, in this transfected cells all have this, we always see that they have pretty mature form of rcDNA. Okay, so their rcDNA inside the cell is already filled and become more mature. And therefore, the precursor, the starting point is different. So that is one possibility. Another possibility is that we found that there's a cytoplasmic nucleus called TREX1. So that is mainly expressed on the ER membrane. And we found that if we overexpress this protein in cell, so then they can deplete the deproteinized rcDNA and prevent the cccDNA to form. However, in the infection condition, okay, so that's the overexpression of the TREX is actually have no effects on cccDNA in the de novo infection condition. So how this happens, right? And this morning I just read a paper that's just post on Baruch, that's preprints websites by Dr. Wenhui Li's lab. That they report that they discovered intracellular receptor now for HBV entry. So we know that virus binding to the NTCP that is expressed on cell surface and then endocytosis. And we actually don't know the molecular detail, how this virus capsid gets out from the endothelial vesicles. So now on his lab found that when the virus gets into the phagocytosis, there is a scavenger F protein that's actually can bind to PS1 region. Because the NTCP binding region is in the N-terminal 47 amino acids. And this scavenger receptor binding site is around 80 to 90 amino acids in the PS region. And this binding actually holds the incoming virus in the endocytic vesicle and transports to the periphery of the nuclear membrane. And that stage, they somehow the capsid is released. Okay, so that's actually perfectly interprets. That's why the de novo infection is not sensitive to tracks. And also, for years, as I just mentioned, that we found that in the cell lines or transfected cells, that's deproteinized rcDNA we detected that is in the first appear in the cytoplasm, right? That means this is at least partially already encoded in the cytoplasm, and then after or express this nucleus, they can be almost completely degraded. So this is actually, in my point of view, is the first strong evidence showing that this cytoplasmic deproteinized rcDNA is actually the real precursor for the cccDNA formation. So that, I think, is a very exciting finding. So then, when the cccDNA already made, is that stable? Yes, it's pretty stable. And thus far, have not found anything that can actually induce a degradation of already formed cccDNA. However, our previous study found that this is a DHBV cell line that can have a lot of the cccDNA copy. So we just label the cell with the fluorescence. That's each cell division that will dilute the fluorescence. So we can use this assay to tracking how many cell division occurs. And we found that each cell division that's you dilute the cccDNA, and in this way you can actually gradually, without new CC formation, you gradually going to dilute and lost. So this is maybe the way that during the HPV infection, that's how you gradually recover. One is totally kill the infected cell. Another one is a surviving cell that can actually dilute out the CCD. And an interesting finding from this study is that interferon actually promotes the core protein decay during the cell division. And as I said, that in the last 20 years, lots of people try to looking for if they can find a compound they can reduce intracellular DNA or cccDNA. So the efforts from the big screening from Roche, they identify these compounds that's found can reduce cccDNA either in these stable cell lines or in the minicircle transfected mice in vivo. However, that's I think how that's actually did it. There published a paper in Journal of Hepatology looks like these compounds induce a lot of the cellular gene change. Okay, I think it's through what kind of mechanism no one knows, but I think that's will not easily to be developed. And the recent work published by in Cell that's shown these compounds. That is an anti-cancer compound that can reduce cccDNA level in the infected primary hepatocytes. And but this compound, so I think it's a cancer compound. It's very toxic. Thus far, I think the most reliable compounds that can reduce cccDNA, inhibit cccDNA formation is actually capsid assembly modulator. So we can see that it's dose dependently reduce the cccDNA in infected hepatoma cells or primary hepatocytes. But how the compounds did it? Because as I said that those capsid assembly modulator is actually promotes the capsid assembly and to form an anti-capsid to prevent pgRNA encapsulation and then prevent CC formation. However, they also have another function. They can actually bind to this already formed capsid. And especially for the mature RC containing capsid, they can make it disassembled. And their DNA contents can be digested by nucleus. So we actually did a genetic study, and we used mutants, core protein mutants. So these mutants, they cannot, binding to capsid assembly modulator, and we can see that wild type you treat with these mutants, you can selectively open up this RC containing capsid, right? After open up, the nucleus can digest it. But this mutant protein, you can see this mature form of the rcDNA cannot be digested. So that means they not open up. Consistently, you can see that, you can see that, the compounds cannot inhibit cccDNA formation when you infect this virus with these mutants, both in hepatoma cells and primary hepatocytes. So then, if we cannot get rid of it, if we have a chance to silence the already formed this CCC mini chromosome. So over the years, many groups, including mine, have tried to looking for epigenetic modulating enzyme inhibitors or something, try to find if that can inhibit the CC mini chromosome transcription. And yes, in cell culture, we do find it. And one most representative results published by Gilead, they found that being a histones inhibitor can potently inhibit the cccDNA transcription in HBV-infected primary hepatocytes, right? However, when they do in vivo test in the UPS kid mice that have hepatocytes, human hepatocytes repopulate, and then infected is actually total lose activity. Okay, so that means you use this in vitro model that may not reflect the hepatocytes in vivo. It's very difficult, have not much thing to get. However, because we know that for cccDNA to when the cccDNA was first made and getting into a nuclear, so our host cell, they do have a defense system. The transcription suppressor complex binding to this cccDNA and prevents its transcription. And then only allows that a small region of the cccDNA to transcribe, that is open reading frame, transcribe the small base protein of the HBV. That long time, we don't know what's the function, we call the X protein. So finally, people figure out this X protein actually recruits E3 ligase and degrades this suppression complex and makes transcription active of cccD. So as expected, some of the srRNA that actually target X coding regions that have been demonstrated that if you use sRNA to knock down the xRNA, so then that cccDNA can be re-silenced. So target X protein that will be a way to silence CCD. People have made tremendous effort to try to leverage this viral protein as a target, have no success yet. Another interesting finding is that interferon treatments actually silence the CCD. How that works? So, here that's, we know that interferon, especially pegylated interferon, is an FDA-approved antiviral therapy. And for a long time, people don't like to use it because it have severe side effects. And however, someone that can tolerate 1 year's pegylated interferon treatment, they do have a chance, 5% chance, that you can actually achieve a functional cure of the chronic hepatitis. So experimentally, when we treat the HBV replicating cells with interferon, we always found about 50% inhibition of viral replication or reduce viral RNA. So, however, in my lab, when I actually did my postdoc work, we produced a cell line that supports the dark hepatitis virus CC replication. And this cell, after we, is a tetracycline useful. So after you remove the tetracycline, they can accumulate about 1,000 copies of cccDNA in this cell. And then in this cell, when we treat with chicken interferon, we see that have a dramatic inhibition of the CC transcription. You see, this is you only need a very low concentration to start work. Okay, and we demonstrate that actually not change the cccDNA level and just suppress its transcription. So use this cell culture system. We did found that interferon actually induced a prolonged suppression of transcription. That means a 24-hour or even shorter interferon treatment. You remove it, and the suppression will last for weeks. And then we did found that interferon induced transcription suppression is related with reduction of the histone active histone transcription marker, like H3K9 acetylation or H3K27 acetylation, but without increase of the transcription suppression marker. So that is a kind of unique. And this finding actually later on was demonstrated by a research group in Novartis. They used HBV infected cell, found a similar conclusion. So then we did RNA-seq and identify about 80 interferon-inducible genes. And then we figure out interferon-induced this suppression must have worked by a nuclear protein. So in this 80-something interferon-induced gene, then we pick up about 20-something, this nuclear protein. And then we did knockdown experiments and identified 3 proteins. So one is that SMAC HD1, another one is PML, and then STAT1. So we did knockdown and overexpression and demonstrates these 3 proteins indeed regulates cccDNA transcription. And then here we also did the epigenetic analysis. We can see that after interferon treatments, H3 K9 acetylation is reduced, right? And also the H3K27 is reduced by the interferon treatments. And when you overexpress these three proteins, so you can see that they also reduce this histone markers. But for each protein, they behave slightly different. If you're interested to read our paper, and then we move on to demonstrate in HBV-infected hepatoma cells, because this SMAC HD1 that is not interferon useful gene in human, so we not test that. Knockouts, the PML and the stats, we all can find that actually, increase the cccDNA transcription. And see, if you knock out, you can see the transcription level is increased. Of course, you interferon treatment, you still can suppress. That means this PML is not solely component. Of course, that one works, but interferon cannot suppress because that was the transcription factor required for interferon signal transduction. So that is the current status. What's our understanding how interferon actually suppresses CC transcription? So now, small molecule cannot work. siRNA works, but you cannot long-term use. And Factor X have not success. And interferon, we know how that works, but we cannot, most people don't like use it because, side effects. So then people think about, gene editing. So actually, lots of labs, including our lab, have been working on develop gene editing therapy to either inactivate or eliminate or transcriptionless silence CCC. Okay, so here is 5 front runner. They have been either is a classic, cut DNA that kind of then let the cell repair it and have some indel and then inactivate CCD. And someone that was actually is use a base editing that actually without cuts, but can actually change the nucleotide sequence, and that inactivates their function. And also, there's artificial nucleus that can actually just like big restriction enzyme, because they recognize about 20-30 nucleotides. You can make it very specific for HBV. So all this is their efficacy have been demonstrated in animal models. At least the 2 have advanced to phase 1 clinical trial. So what's their efficacy? We will soon know it. Okay. And Dr. Ju-Tao in our group that actually worked with Minhong in a company in our building that's developed RNA editing technology by focus on optimize the guide RNA. We know that for this gene editing, you need a Cas protein. You also need a guide RNA. So the current guide RNA is about 102 nucleotides long single-strand RNA. And the manufacture is an issue, and also you cannot optimize it because your total synthesis is pretty difficult. And Minhong developed a method. You can actually use non-nucleotide chemical and to in this loop region and make it called chemically ligated guide RNA. So right now, we demonstrate that you can either make it 2 segments ligated or 3 segments you ligated. We call it LgRNA or L2gRNA. And Chang's experiments actually shows that all this worked. Okay, and however, side-by-side comparison with sgRNA, they do have some advantage and disadvantage. However, that gives us a platform you can chemically modify it. Because this is very important, because previously you need to synthesize a single-strand RNA, 102 nucleotide long, then now you can synthesize about 30 nucleotides and then 3 segments you are getting. And another advantage is that for difference, guide RNAs, the only sequence difference is the cRNA that recognizes target sequence. So 2 segments you can actually make a lot and only change the cRNA sequence. So this is thing we are currently work, and hopefully we can soon demonstrate its efficacy in vivo. So then my presentation is roughly, so that's scientific content there. And then think about what's a standing question. I think that chronic hepatitis B, as I said, that's affect about 250 million people, and even have a very effective vaccine. So each year still have a lot of people continue infection. So I think the disease burden is very big, and also the long-term consequence is very bad. About 1/3 of those infected chronic carrier were developing into end-stage liver disease, like cirrhosis or liver cancer. So the treatment is really need to improve, because currently we have nucleoside analog viral polymerase inhibitor that's very efficient inhibit this reverse transcription state, right? And can inhibit this state. And they can reduce virus logs by 6-7 logs. And after 1 year of treatment, more than 80% of the people, their blood virus cannot become undetectable. However, because this cccDNA is very stable, seeding, and after you stop the treatment, and then replication going to come back. So you can see why the cccDNA is so important, okay? And also another barrier is that, is our immune system. So normally, when this virus infected adults, so over 95% of people that will resolve this virus infection within 6 months. And when the virus infected neonates, just a newborn baby, this kind of resolution, immune resolution of infection, the chance is only 5%. So most majority of neonates infection has become chronic carrier. And then a very important question is that, so now the virus expresses 7 protein, and we have a functional immune response. In the HBV carrier, they have a normal immune response to defending other viral infection. Even for example, like dengue virus or something, they also infect the liver, right? So in the epidemic region, so they resolve the dengue infection normal. So then why they cannot mount efficient immune response to resolve the chronic HBV infection? So the immunological basis until now, we know something, but when we know more, we have more questions, okay? So it's still need to work out, right? So I think, in my point of view, for cure chronic hepatitis B, one, we need to further understand cccDNA biology. We try to destroy it or silence it, okay, to achieve a sustained response. Another one is that actually we need to understand our immune system in the chronic carrier. What's wrong? If we have a chance actually to jump it up. So I do think we have a chance because some of the chronic carrier, they do automatically without treatment. After years, the immune system wake up and resolve the infection. Okay. So right now, I think that's also is a very important area. We try to understand the immunopathogenesis and try to develop immunotherapy to control the chronic HBV infection. So here, so finally, the work, I present, that's not only just my own work, I put in the context of the work in the whole field. So for my own work, this is the current my lab member, and this is for many, many years. So all the alumni of my lab in Drexel or Blumberg, that's contribute. And also, oh, sorry. Oh, that's a duplicate. So I put a wrong slide. So then our colleagues, that's like Dr. Chang, Dr. Du, that's our long-term collaborator for decades, okay, and other people. So finally, that's really appreciates a man that's really makes this place different. That's has his name on our building, Dr. Block, the founder of our institute. So thank you. I stop my presentation here, and any questions?
I have a question. Do HPV-infected hepatocytes express any particular specific antigens on the cell membrane?
Yes, that's but somehow, I think the major issue is that in the current carrier, if you want to find HPV specific CD4, CD8 cells, the number is very you need to take the blood and stimulate with peptides for 2 weeks, and then you may see small amount. This is not like you infected with the flu. That's in short period of time. Close to 1% of your T cells is a flu specific, right? So I think it's in the current carrier, the T cell. That's either the number is very small, and then when you test their function is also looks like the cytotoxicity activity or secretion, the cytokine, the ability is all reduced. Yeah. So that kind of immunotolerance stage, how that's achieved. And also, why in a small fraction of patients, they actually automatically wake up, right? So I think, this is a chance, just a case, compared to HIV. There's never have a chance to self-resolve, right? And but for HPV, you do have every year, it's 1% per year, that kind of chance for self-resolution. So I think, that means our immune system is not functional, but not totally bad. We just need to find a way to, activate it in most of the people. Yeah.
So Ju-Tao, are there any enzymes that can degrade double-strand linear DNA?
So I think, inside the nucleus, the DNA degradation should be very strictly controlled because, if you have a nucleus in the nuclear, how you can actually, distinguish what is your chromosome DNA, what's your, viral. So we do have lots of nucleus, especially, those nucleus actually involved in the DNA repair. But that nucleus generally is well controlled. That's in a certain kind of multi-protein complex, right? So basically, you do have nucleus inside a nucleus. So how are you going to make it specific? So actually, gene editing is a nucleus. So that Cas9 is a nucleus. So that cleavage requires the guard RNA to guard the nucleus to the cccDNA. So I think, that's, oh, that's wrong. Yeah, that's a way people doing it. But I think, how we immobilize the host cellular function to get rid of cccDNA. Because, there's a lot of the knowledge gaps.
Those two structures you showed are sort of different from each other structurally.
Yeah.
Does that mean they have different mechanisms of action?
The two different species of the DNA.
Oh, they're not the same.
What?
The two different chemical structures that were inhibitors of the.
That's totally different. Okay. So one Roche compound, you actually don't know what they do, but that's recent report that that compound actually is a known compound, is an anti-cancer compound that's actually is a DNA chelate. They can directly bind into DNA. So I think that's you don't have much chance to use that one as a therapeutics.
I understand.
So Ju-Tao, we found out that the Roche compound is a sensitizer, so it elicits some kind of allergic reaction in humans. So it's possible that compound is acting through some kind of a immune pathway as well.
Yes.
So it's pretty toxic compound.
Yeah, based on what they published in that's journal hepatology paper. So they did RNA-seq. They detect a lot of transcription change. So that's actually is a signal. Those compounds either pretty dirty. They do a lot of the things off-target effects or something, or they reduce cccDNA, nearly immobilize a lot of the host function, right? So I think as a chronic disease, and those people already have a bad liver. It's not easy to tolerate. And
Just one last question. I mean, aside from that fact that Roche compound, nobody can reproduce Roche's results from. That's the scuttlebutt butt in the industry. But what do you think? What's known about possible biological functions to secreted enveloped empty capsids and RNA containing particles? The RNA one's position was that maybe they were actually still infectious, but
no one actually clearly demonstrates that RNA containing virus is really infectious because its number is much smaller than the DNA containing particle. So I think the virus in many ways looks like they are very smart. They have done something, pyramids, lots of the host function, and just for their own purpose. However, sometimes you will find that they also do something pretty sloppy. Because this not like the DNA replication, all that things, you have lots of the proofreading. So viral particle assembly, that's macromolecular assembly process. They don't have too much proofreading. Okay. So that means they can assemble something and they failed, right? Because they made a loss, right? So I think, it's, that actually is for a lot of viruses, that is true. think about the influenza virus, right? There is a lot of the defective viral particles there. And actually, how interferon was discovered because study this defective viral particles. Yeah.
Is it possible to predict who's going to get liver cancer based on a molecular profile, based on a pattern of infection?
So I think the one factor is genetic. That's why current clinical guideline is that that's if you contract have a chronic carrier of the HPV. So for most majority people, the antiviral therapy, they want to start when you really have a liver disease like hepatitis, your ALT is elevated. Let's start fit. Of course, the current the guideline already rules, but previous that's really if you, however, if your family someone have a liver cancer, so then you need to treat as early as possible. So that's and also there's a lot of the genetics change may related, but not like colon cancer, breast cancer. That's cancer genetics work have been very advanced, but unfortunately for liver cancer, there's the understanding is still limited. However, last year a Chinese group that have been did the deep sequence, whole genome sequence, RNA sequence, all those omics studies about 500 HPV related liver cancer, and discover a lot of very interesting genetic change that's related with HCC oncogenesis during chronic HPV infection. That's, I think, going to be a very rich source, so we can further explore it. So otherwise, I think that's our third program called Precision Medicine Program. Our efforts is actually develop those DNA, cell-free DNA or RNA-based assay and do the screening. So that's in reality, we're not going to achieve the early diagnosis, but is a cancer risk evaluation assay. So if you have certain marker light up, that means you have a greater chance to have a cancer. So that's right now, I think, is practical level. We try to do this early diagnosis, and but because the HPV chronic infection, but like I said, that's despite you have 1 out of 10,000 chance for the HPV DNA integration in a single cycle infection. However, chronic carrier for several decades that can really accumulate a lot of the integration, and you don't know which integration that will hit the jackpots. That's inactivates your tumor suppressor gene or activates the oncogene. So I think that's whatever, even just for suppressed viral application. I think the treatment should be start as early as possible. That's, I think, is the best way to prevent people develop into the liver cancer. Okay, great. Thank you.